Structure of protein. Now after understanding the amino acids, now let us first understand how amino acids joins to each other or combines with each other to form proteins. Now what you need to know is, we write amino acids like this. It's R1 COOH. So this is the first amino acid, well, the one amino acid we are having. And let's take another amino acid which is given by R2. Now, when these two different amino acids, they combine with each other, what happens is, loss of water molecule take place, minus water. And this is how R1 C double bond O NH CHR2 COH, this is bond. So this amide group, which is responsible for joining the two amino acids, is called peptide bond. So remember, in proteins, amino acids are bonded to each other through a bond which is called peptide bond, chemically which is an amide group, formed by the loss of water molecule between the acid and the NH2 group of two different amino acids. The examples can be, it can be glycine, it can be alanine, and what we can get is the combination of these two amino acids. Now when the two amino acids combine to each other, it's called dipeptide. When it combines with one more amino acid, it forms what? Tripeptide. And like this, it can combine with many amino acids. AA denotes amino acids. It forms a long chain which is called polypeptide. So what is polypeptide is? Polypeptide is a linear polymeric chain of amino acids in which contains one amino acid after the other and so on. So long chain is formed. When these amino acids are more than 100 in number, we call it maybe a protein. See, there is a very thin line between a protein and a polypeptide. Sometimes a normal polypeptide chain which can exist independently can also be a protein, and sometimes what we find is that even this normal polypeptide chain, which is very long one, which can exist but is not regarded as a protein because it has to exist in a very complex structure. Like example is insulin, which contains a normal 51 amino acids, is an independent a protein. That's actually is a linear chain of the 51 amino acids. So that is how the complexity is. But for what we have to understand is this peptide linkages and how this polypeptide chain is formed. Now let us understand the proteins. Proteins, as per their molecular shape, are of two types. On the basis of molecular shape, proteins are of two types. One are called fibrous protein and other are called globular protein. Fibrous proteins are long linear chain polypeptides of polypeptide chains which are held together by strong hydrogen and by disulfide linkages. Now these are very strong, very strong because they are strong, held by very strong linkages, having very high tensile strength, are water insoluble in nature. They are fiber-like, that is why they are called fibrous. And the main examples of them are the keratins, which are there in our hairs, or myosins, which are there in the muscles. Now, when the polypeptide chain is existing in the coiled structure like this, it takes the shape of a globule and it's called globular protein. Such type of proteins are having weak or different types of forces of attraction between the polypeptide species and are water soluble. The best examples to explain globular proteins are your albumin. In the albumin we find, and we have insulin. So these are the proteins on the basis of their molecular shape. Now let us understand the structure and the shape of the proteins. The structure and the shape of the proteins is studied in 4 stages called primary, secondary, tertiary and quaternary. So let us first study the primary structure of protein. The primary structure of proteins means how the amino acids are arranged in the polypeptide chain. Remember, primary structure of a protein means how the amino acids are arranged in a polypeptide chain, because that sequence is very important, and once that sequence is changed, the polypeptide's nature is completely changed and the protein or the enzyme which it will be forming will be completely changed. So I can give you this structure like if we have CHR1 C double bond O NH CH R2 C double bond O NH. So it's like this. So this polypeptide chain is jigsaw puzzle, like this is zigzag, it's like a zigzag chain in which we are having what one amino acid lying one after the other, and this is how the long chain formed are called the primary structure of proteins. Then is the secondary structure. Secondary structure of protein it means how the polypeptide chains arrange themselves to the next level. How they arrange themselves to form the higher complexity level is called the secondary structure of protein. Now, polypeptide chain arranges themselves in 2 possible ways. That is why secondary structure of proteins is of 2 types. That's called alpha helical structure or beta pleated structure. Now, what is this alpha helical structure is? When all the NH group and the carbonyl group of the polypeptide chain are engaged in themselves only in forming the intramolecular hydrogen bondings, like that the intramolecular hydrogen bonding, and they forms a right-handed helical structure. It's like this. This is what the right-handed helical structure we have, where what we find is the NH group is bonded. The NH group is bonded through hydrogen bonding to the carbonyl group of the next turn. Like this, the NH group of this turn again is bonded to the carbonyl group of the next turn. And this is how what happens is they get coiled up to form right-handed helical structure. So all functional groups are actually bonded to each other through hydrogen, intramolecular hydrogen bonding, and therefore they attains at the end a long polymeric or a long fiber-like structure, which are called alpha helical structures. Whereas, what is beta pleated? These are open or expanded chain structures in which one chain lies just after the other, and they are arranged in such a way that they takes the shape of the pleats which we have during in draperies or in the curtains or curtains. That's why they form pleats one after the other. That's why this structure is called beta pleated structure. In this, one polypeptide chain is actually attached to the other polypeptide chain through intermolecular hydrogen bondings and the disulfide linkages, etc. But what we have is, since the chain structure is open and they are lying one after the other, it's called beta pleated structures. That's the secondary structure of protein. Now, after this understanding secondary structure, let's move to the quaternary structure of proteins. But before this, let us first understand tertiary structure. See, we have seen in the secondary structure, the protein polypeptide chains arranges themselves in either in the alpha helical manner or in beta pleated manner. Now, how these arrangements are again further combined with each other to form another next higher level of protein is called tertiary structure. Now, the tertiary structure of proteins is of two types. It's called fibrous and globular. We have already discussed about it. Fibrous, yes, now we can say is only made up of what the alpha helical secondary structure proteins. So it is actually having a fiber-like. So all these fiber-like polypeptides arrangements which we find are basically secondary alpha helical structures. That is why these proteins are water insoluble, are having very high tensile strength because of the strong hydrogen bondings, and they are fiber-like. Whereas globular protein is mixture of both fibrous as well as globular protein. So it's a mixture of the alpha helical as well as beta pleated structures. Since groups are in the beta pleated are free, which makes these proteins water soluble. So this is how we get fibrous and globular type of tertiary structures of proteins. Then let us move to the last stage of quaternary structure of protein. See, in many cases what we find proteins last structure is either globular or fibrous. But what we find in some cases that some proteins are made up of two or more types of different polypeptide chains. Now in their structures what we find that their spatial arrangements are different, and those spatial arrangement structures are called quaternary structures of proteins. So this is what is the complex level of the structures of proteins we have.